Let us begin the new chapter, Respiration in Plants. Students, what if I ask you what are the three primary requirements of a living organism? Definitely your answer would be air, water and food. We need air for the oxidation of food. Food gives us energy and water is the universal solvent in which all the reactions of the body are carried out. Now, for the animals, rather higher animals, they have a well-defined organ system to provide oxygen to the body. This is because they have a very high energy need, especially homeotherms, which can maintain a constant internal environment. They have a huge oxygen demand, so they have well-defined organ system. But think of plants. They do not have any well-defined organ system for provision of oxygen in their body. This is because the requirement of external oxygen by the plants is very limited because of their limited energy requirement. They undergo a very less amount of oxidation of food due to their relative simpler structure. So, no need of a specialized organ system. Moreover, in the leaves, they have plenty of air spaces through which air can diffuse inside the cells. The stems have lenticels for respiration. At the same time, as we know, the plants photosynthesize, so they produce rather oxygen as the byproduct which is utilized in the process of respiration. The food required by plants similarly is very limited. They are, along with the cyanobacteria, the main producers of food on the earth. It is because of them others survive. So they photosynthesize food that we know very well. The animals are heterotroph, which derive their food from the plants in the first level, and then we have carnivores which derive their foods from herbivore. Ultimately, we all depend on plants for our food, as because we cannot synthesize our food. So the requirement of food by animals is huge one as compared to that of plants. And water, as we all know, is required to carry out the various reactions. It is also a reactant in many reactions in the body. Now, for respiration basically constitutes of two parts. The initial part is called breathing, as you all know. You have studied in class 10. What is breathing? The physical process of taking in oxygen and giving out carbon dioxide. And respiration, we mainly refer to the cellular respiration, where oxidation of food releases energy. Energy in the form of high energy bound molecules called ATP, the adenosine triphosphate, the energy currency of the cell. These ATP molecules, they have energy rich phosphate bond, which on dissociation produces or releases huge amount of bond energy, which is utilized for various purposes in both plants and animals. Now, for this, oxidation of food is required for the production of ATP. The oxidation of food occurs in two steps. The primary step or the first step is glycolysis, which is common to all types of organisms. The second step depends upon the availability of oxygen. There are basically two types of organisms. Aerobes, which require oxygen for the oxidation of food, and anaerobe, which do not require oxygen for the oxidation of food. The anaerobes or aerobes, they can be facultative or obligatory. The facultative anaerobes are the organisms which can oxidize food both in presence and absence of oxygen, like your E. coli, Escherichia coli. And obligatory anaerobes, they strictly require absence of oxygen for the production of energy, as Clostridium. Clostridium tetani, you have heard about, the causative organism of tetanus. It is a obligatory anaerobe. Now, as I said, the first step of oxidation of food is common to all organisms, and it is known as glycolysis. Three scientists, Emden, Meyerhoff and Pernas, they found out this particular pathway, and that is why it is also called EMP pathway. Emden, Meyerhoff and Pernas pathway. So, what is this glycolytic pathway like? Let us see in details. The glycolytic pathway occurs in cytoplasm of the cell. So, be it prokaryotic or eukaryotic, they all have cytoplasm. So, they all carry out glycolysis. So, glycolysis starts with glucose, and it is a 10-step process. So, the glucose first undergoes phosphorylation to form glucose 6-phosphate. This step is catalyzed by the enzyme hexokinase. So, a phosphate group is required for glucose. So, that is given by ATP. The ATP forms ADP, and the released phosphate group is incorporated into the glucose molecule at the 6th position. So, glucose 6-phosphate. This glucose 6-phosphate isomerizes into fructose 6-phosphate. You must be knowing glucose and fructose both are 6-carbon compounds or 6-carbon sugar. So, glucose is an aldose sugar and a fructose is a ketose sugar, and they are isomers of each other, having the same chemical formula but different structural formula. So, glucose 6-phosphate isomerizes into fructose 6-phosphate by the enzyme phosphoglucose isomerase. So, you have studied in enzyme chapter that it is the isomerase enzyme which isomerizes the substances. So, here phosphoglucose isomerase isomerizes glucose 6-phosphate into fructose 6-phosphate. The fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. Means another phosphate group is attached to the fructose 6-phosphate. So, this step is catalyzed by another kinase enzyme, that is phosphofructokinase. And since phosphorylation occurs, we need a phosphate group that is donated by ATP. So, fructose 6-phosphate gets converted into fructose 1,6-bisphosphate by the enzyme phosphofructokinase. And ATP gets converted into ATP, donating its phosphate group to the fructose 6-phosphate to form fructose 1,6-bisphosphate. Now, this fructose 1,6-bisphosphate is a 6-carbon sugar with 2 phosphate group at 2 ends. So, that breaks down from the middle by the enzyme aldolase to form 2 compounds. One is called glyceraldehyde 3-phosphate and the other is dihydroxyacetone phosphate. Both of them are 3-carbon sugars or trioses. So, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are formed from fructose 1,6-bisphosphate by the enzyme aldolase. Now, these two are isomers of each other. This is a aldehyde sugar, this is a ketone sugar. So they can be isomerized by the enzyme triose phosphate isomerase. So the fifth step of glycolysis is the isomerization of dihydroxyacetone phosphate to form glyceraldehyde 3-phosphate. So students, see, from one molecule of glucose, say one molecule of glucose, we ultimately get 2 molecules of glyceraldehyde 3-phosphate. Now glyceraldehyde 3-phosphate undergoes dehydrogenation. It undergoes dehydrogenation to form 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. And who is the hydrogen acceptor here? Dehydrogenase means hydrogen is going out. So somebody has to accept that hydrogen. Who accepts that? NAD. So there are 2 molecules of glyceraldehyde 3-phosphate. So we require 2 molecules of NAD plus to form NADH, and in turn glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Now this NADH, it moves to the electron transport chain to form the ATP. Remember, a single NADH molecule forms 3 ATP molecules through the electron transport chain. Now this 1,3-bisphosphoglycerate gets converted into 3-phosphoglycerate. So how it will form? It will give up one phosphate group. So 1,3-bisphosphoglycerate gets converted into 3-phosphoglycerate by giving up a phosphate group. And who takes up that phosphate group? ADP. So from ADP, ATP is formed by accepting that phosphate group, and the enzyme here involved is the phosphoglycerate kinase. So phosphoglycerate kinase converts 1,3-bisphosphoglycerate into 3-phosphoglycerate, donating a phosphate group which is taken up by the ADP to form ATP. So as I said, one glucose molecule yields 2 molecules of glyceraldehyde 3-phosphate. As a result of 2 molecules of 3-phosphoglycerate would be formed, forming 2 ATP molecules in this step. Now 3-phosphoglycerate gets converted into 2-phosphoglycerate by the enzyme phosphoglyceromutase. So it gets the phosphate group from 3 position to 2 position. So 3-phosphoglycerate gets converted into 2-phosphoglycerate by the transfer of a phosphate group from 3 position to 2 position brought about by phosphoglyceromutase. So 2-phosphoglycerate then gets converted into phosphoenol pyruvate by the enzyme enolase. A water molecule is given out. The phosphoenol pyruvate again gets converted into pyruvate, the ultimate product of the glycolytic pathway, by the enzyme pyruvate kinase. So the phosphate group in the phosphoenol pyruvate is donated by the PEP to the ATP. As a result, ATP is formed. So again, we had 2 glyceraldehyde 3-phosphate, so 2 phosphoenol pyruvate, which will form 2 pyruvate molecules, along with it 2 ATP molecules. This step is catalyzed by pyruvate kinase. So students, these 10 steps, they bring about the breakdown of glucose to pyruvate. Now fate of pyruvate depends upon the availability of oxygen. What is to be noted here is, see, the first step is not reversible. Step number 1, step number 3 and step number 10 are not reversible. all other steps in this process are reversible. Number 1. Number 2, see the energy kinetics of glycolysis. What happens here in this particular step, your 2 ATP are formed. Here also 2 ATP are formed. So there's a net gain of 4 ATP in the glycolytic pathway for one glucose molecule. But here we have used up 1 ATP. Along here we have used up another ATP molecule. So 4 ATP gained, 2 ATP lost. So there is a net gain of 2 ATP in the glycolytic pathway. So I hope it's clear that this glycolysis is the primary step of oxidation of food. One molecule of glucose yields 2 molecules of pyruvate. This pyruvate, or depending upon the availability of oxygen, either enters into aerobic respiration or anaerobic respiration, or may be used in lactic acid formation in case of constraint of oxygen. And then there's a net gain of 2 ATP molecules in this process. So in our next lecture, we will see the TCA cycle, or how this pyruvate is used up for the energy production.